At 5.
Oh, they are. So everything's in this Monday session. Guess we should get started with our second session today. And the session 2 is the early phase of drug discovery. So this morning you've heard some very nice talks on the various aspects of medicinal chemistry as well as small molecule medicinal chemistry as well as natural product medicinal chemistry. And so this session will kind of shift the equation a little bit from the medicinal chemistry side of the collaboration that is required in drug discovery and chemistry and pharmacology to more of the pharmacological aspects of the early stages of drug discovery. And so we're going to have 3 speakers today. And you know, as we think about the presentations that are upcoming, I just kind of put this cartoon together, similar to what many of you have probably already seen in various other slides of this kind of funneling process of the drug discovery process, where you start out typically with a large number of compounds in a compound library. The number can be anywhere from 50,000 to 1 million, depending whether you're working in a small academic lab to a pharmaceutical company. But of course, it's a process of attrition, where essentially you do your high throughput screening, and if you're running a screen that's kind of typical, you might get a 0.1% hit rate, so that narrows this all down to about 500 compounds, where you want to, of course, confirm hits and do dose response. And so this process of the high throughput screening is going to be covered this morning then by Jim Inglese, who's at the NIH Chemical Genomics Center, and he'll give a nice overview, I think, on the basics of high throughput screening. Now, once you've completed the high throughput screening process, done hit confirmation, hopefully got some signs of dose response of your compounds, you may be down to a smaller number. In other words, there's obviously attrition just in the confirmation and dose response testing. But let's say, for argument's sake, you're at 100 compounds, and this is where you start now looking at the features of these early lead hits, including things like selectivity testing. So if it's receptor A, let's say a serotonin receptor, you want to look to see if it has activity at related serotonin receptors. And you'll start essentially honing in on ideally preferred, let's say, scaffolds within these hits. And ideally, you want to have at least a couple scaffolds that you might select to go forward into the analog synthesis phase, where now you get the medicinal chemistry we heard about this morning entering into the process. And whether they make 30 compounds or 150, as we heard, is a little bit dependent on the target and dependent on the resources that you're dealing with. But this is the iterative process that we heard so much about. So you get the analogs, you test them again for potency and selectivity, and you go through this loop trying to refine these 2 endpoints. But there's also some iteration downward in the sense that ideally you want to look at some of these other properties that were alluded to this morning. So the ADME properties, whether they essentially have good solubility, whether they have reasonable behavior as far as brain penetration, et cetera. And I'll cover in my presentation after Jim's some of these aspects of compound optimization after HTS. And then finally, if you're really lucky, you continue to triage compounds. You start to refine even more in this process. You find compounds that have good pharmacokinetic and ADME properties. Hopefully maybe some preliminary safety suggests they're good. You move a couple into in vivo efficacy testing. And if you're real lucky, and you get some positive results, and you might even have something you consider going into the clinic with. And so Dr. John Sagertz will give you an overview on some of the importance of the toxicological studies that will be needed to allow you to get into essentially, ultimately, if you're, an IND type path into phase 1 clinical testing. So with that, I will turn it over to Dr. Inglese, who will talk about high throughput screening.
Thank you, Kurt, and I'd like to thank Howard and Kurt for inviting me here today. So I just want to check, do I get 20 minutes or 30 minutes? 20? Okay. Well, this is not. So a quick outline of what I'm going to be discussing today on the slide. I'm going to go through an overview of the high-throughput screening process, and it currently is practiced across the majority of industry and academia. then talk a bit about the chemical libraries, the spectrum of chemical libraries in use today. And the design, this is going to be an important component of my talk, the design of assays and how they need to be compatible with this infrastructure we call HTS. And then issues that arise at the intersection of the chemical libraries and the high-throughput screening assays, very important. And hopefully to illustrate some of these points, I will have 2 case studies, one on peripheral neurological systems and one on central nervous system disease. And then hopefully I'll conclude with a little bit on what the NIH is doing to help facilitate drug discovery in these areas. So the high-throughput screen is basically an iterative testing of different substances in a common assay, generally for greater than 10,000 samples per day. This can get much higher than this. The system you saw, if those of you noticed the video playing, that wasn't Merck or Pfizer, that was the NIH's robotic platform for doing high-throughput screening. These are becoming ubiquitous throughout the country, both within and outside of industry. So the assays are designed for high-throughput screening. They attempt to integrate the biological fidelity of the with high sensitivity assays and the screening technologies. Now, the configuration and nature of the assay formats are critical to the HTS experiment, and they must be coordinated with the nature of the compounds tested, okay, and the subsequent assays that are used to evaluate the biological relevance or mechanism of action. So it's not just that developing a high-throughput screen isn't just making one assay. It's a whole portfolio of assays that have to be coordinated with that. Now, in order to do these types of experiments in very high throughput in the numbers you just saw, we have to really work in low volume formats. And currently the field focuses around either the 384 well plate or the 1536 well plate. I'm sure everybody here has worked in the 96 well plate format. But so these are the plate formats. You can see the footprint's identical. However, we have many more wells in this high density 1536 well plate format. This means a lot of things. First of all, that means you really have to know your biology quite well, and you have to use the latest technology to integrate it into a low volume assay, and I'll go over a little bit of that in a minute. The volume of these plates is on the order of 4 microliters as opposed to 50 for the 96 well plate. Now, one of the advantages of a 1536 well plate is you have a lot of real estate now for control. So there's 128 control wells in such a plate. If you take what was equivalent in percent of the plate for that was for the 96 well plate, you only had 8. This is very important, especially when you're trying to optimize and evaluate the data coming out of a very small micro well. Now, just to give you an idea of how parallel processing in this manner can really expedite the screening process. So for a 200 plate run, say over 24 hours, you can see the time that it would take to screen 96 well plates versus a 1536 well plate format. So for 1 million samples, you can do that in 3.5 days as opposed to 3.2 months. Now, to place reagents into these small wells, one has to have sophisticated liquid dispensing and compound dispensing technology. And those are primarily for the buffers and cells that are added. These are pressure actuated microsolenoid dispensing systems shown here. For compounds, it's been traditionally they've been traditionally added by this pin tool array. So it's 1536 pins that dip into a compound plate and then into an assay plate. There's issues with this. It's very fast because it's a simultaneous transfer of 1536 or 1408 samples, if you discount the control wells, into an assay plate. However, there's potential carryover. We've worked that out pretty well, though. But we're now replacing this with acoustic technology, which can use sound waves to eject small volumes from DMSO-containing microtiter plates into your assay plate. That's much slower at the moment because it's usually a single transducer. This is an example of that down here. You, of course, have to flip your plate, your recipient plate, upside down over the compound plate, and then an echo comes through and blasts out the sample. And 1536 well plates are no problem to flip upside down because the surface tension holds all the solution in the well quite nicely. Now, of course, at the end of the experiment, after all the reagents are added, be these cell-based assays or biochemical assays, one needs to make a measurement. Generally, we're making optical measurements using multimodal detectors. As you can see here, we can usually do anything from absorbance, and this is all possible in 1536 well format. Bioluminescence is one of the most widely used and sensitive outputs, and fluorescence, of course, is also used. There's a lot of progress being made to actually move gold standard assays such as patch clamp technology into microtiter plates using the planar patch clamp, so we can make current measurements actually on cells in microtiter plates, and you can see the type of output you get. As well, of course, automated high-content imaging to look at basically bring microscopy into the realm of high-throughput screening. So all of this is possible now. And of course, what you need to do with that complex output is distill it into an idea of the compound activity versus the compound itself. Now, so what are the types of libraries that we can interface with this type of technique? Well, we've heard quite a bit about chemical libraries so far. So library membership, of course, ranges from very small to moderate to large, hundreds of thousands or several million samples. And they can be classified broadly. I have them classified broadly as either biologically active, and we know that these are very limited in number relative to those that are biologically uncharacterized. We heard the numbers earlier today. These are vast. So the trick, of course, is if you're working with biologically uncharacterized libraries, such that you would buy from Cambridge or somebody like that, these are typically low diversity, high density type libraries, such as CombiChem libraries, or consolidated sample collections with extensive structural diversity. One has to decide what you're going to screen here. Clearly, if you're working with these very large libraries, you have to have the high throughput capacity that you saw on that movie. However, if you're working with smaller size libraries, one has more flexibility in the type of automation you might want to use. I should point out that we heard a bit about natural products. So biologically active libraries can compose complex mixtures, natural product extracts, or pooled synthetic libraries, or even synthetic extracts are being discussed now in the literature. And of course, there's privileged scaffold-based libraries which take these molecules of known biological activity, make analogs without actually knowing what they're going to do. But for example, the imidazyl pyrimidines, these are kinase inhibitors based on that 38 compound discovered at SmithKline. Now, here's some specific examples of libraries. Everybody's probably familiar with the LOPAC or the Sigma library of pharmacologically active compounds. It's used in our lab now as just kind of a validation library for any assay we develop. But they range from libraries that are called natural product compound libraries from companies like Timtech. There's even libraries of toxic compounds that we have at NIH for the National Toxicology Program. I'm highlighting two here because these are going to be in my case studies. The molecular library, the NIH Library Small Molecule Repository, or LMSMR, is a rather large 400,000 member library, and this is one of these diverse collections that Chris actually helped in the early days NIH put together. It's outsourced from different vendors, and it's very highly QC'd, so we're very certain, as best we can be, about what's in each well. And I'm also going to talk about a collection that we just put together at NIH, which is called the NIH Pharmaceutical Collection, and that's approved drugs, all approved drugs, not just in the US, but also elsewhere in the world. And this, of course, is for repurposing. And so it's if you look at the number, it's a very relatively small number of compounds, about 3,000. So that's pretty much the universe of approved drugs. Okay, so the design of the assays is probably, to my mind, one of the most critical aspects when you're thinking about high-throughput screening. And I define the assay as an efficiently designed experiment measuring the biological effect of a substance, the effect of a substance on a biological process of interest. And it's kind of this chicken and egg thing. You really almost never start off with the right HTS assay. It's iterative process. So this is actually a rooster, and I don't believe they lay eggs. So you can think of your first assay in this manner. It's probably not going to necessarily give you what you want, and you need to tweak it and work on this. And if I had more time, I could explain a number of examples of this. But eventually, you want this rooster to become a chicken and lay the golden egg that we are always all looking for. But I think I would like to just point out that you have to really spend the time to develop the right assay or assays, because I think, as we've heard, the cost of failure appears to increase exponentially the further it occurs from the start of the program. Many investigators don't want to spend a lot of time developing assays because it takes sometimes quite a bit of molecular biology or getting the proteins purified, but it's really worth the effort. Now, some of the special requirements of high-throughput screening assays are in this table. All of what I'm discussing has been published in some reviews by my colleagues and I. They're down here. Just point a few things out. For example, reagents. Often, for the benchtop assay, often are limited in quantity. They vary in batch. And for high-throughput screening, we really have to have a standardized batch of material. Here's an example. Here's a stable clonal cell line in a 96-well plate expressing a firefly luciferase. And here is the same plasmid transiently transfected. And if you're sitting in the back of the room, these probably look pretty equivalent, and you might be able to get away with transient transfections if you're doing 96-well screening. But what often happens, you take that same plasmid and you compare the clonal versus the transfected in 1536, and this is what you see. So, batch stability, clonal lines are almost always critical in high-throughput screening. We're trying to improve the ability to use transient transfections, but that's all part of the whole research in applied science of high-throughput screening. One of the other things I wanted to point out is the protocol. Very important to have a streamlined protocol. So you can imagine protocols in the normal lab can be complex, many numerous steps, separations. But in the high-throughput screening environment, we really like to keep that to a minimum number of steps. So here's an example of some of my work when I was a postdoc. Now, this is a complex assay to measure the phosphorylation of light-activated rhodopsin. And it may be good enough for a Nature publication, but this will no way be suitable for a high-throughput screen. There's Eppendorf tubes, vortexing, centrifugation. You've got to go into a dark room. Clearly not tenable. This is more like the type of protocol one needs to design for a high-throughput screen. And I'm going to talk about both these examples in my remaining time. For example, here, some work we did with Kurt and his colleagues at Penn, a biochemical assay that looks at tau fibrillation. And you can see the protocol is just 9 steps, 2 of which are just detector reads at the end. So it's a 7-step protocol. And then one I'm working on currently with the Charcot-Marie Tooth Association that's more of a phenotypic screen. And this is a cell-based assay with 7 steps. And again, you can note the volumes of these assays, 2 microliters. And then we had another 2 of reagents. So it's about a 4 microliter assay. And here it's about a 4 microliter assay as well. You might have noticed that I don't think I need that. I have the concentration of the compounds tested as a range of concentration. I want to get into this in a minute. So technologically enabled high-throughput screening creates an efficient interface between biological assays and chemical libraries that allow the rapid identification and profiling of wide-ranging chemotypes that modulate the gene, product, or cellular organs and phenotypes on a large scale. And so this is the simple equation. We have our assays appropriately designed in our chemical libraries. We can automate this, use informatics to help process the data, and we hopefully will have the goose that lays our golden egg. And those of us from the East Coast who live in locations where there's grass on the, that we walk across, will realize that on average, your typical goose does not lay the golden egg. There's other material that this fowl will produce. And this is not unlike high-throughput screening. I'm sure Chris would agree with this. Try to hold your applause. And this is the Achilles heel of high-throughput screening, actually. You can generate a lot of data, a lot of information, but what do you do with that? And that's the key to success or failure with this type of technology. Let me just talk about this briefly. I've spent a lot of time, I essentially made a career out of studying the artifacts that come out of high-throughput screening, and as well as some of my colleagues. Brian Schoikett at UCSF has done beautiful pioneering work to understand how aggregates affect biochemical assays. I've spent a lot of time looking at reporter gene phenomena, especially with the luciferase enzyme. I don't have time to go into it now, but you can get confounding, counterintuitive outputs based on the pharmacology of the library interacting with the reporter as opposed to the biology. So, for example, just quickly, the aggregation phenomena, the compound can aggregate certain types of compounds and appear as an inhibitor because it's sequestering your enzyme. and denaturing it. But if you were to add detergent and basically capture the colloidal suspension, the activity would come back. So that's one diagnostic, for example. And then there's redox cycling activity when DTT is present with some compounds, et cetera. So here's what you do. If you're this fella and you're trying to catch a fish, he's going to what? Is he going to be successful? Probably not, right? But if he knows something about the refractive properties of water and Snell's law, he can make some compensation and do a little work, and he'll get his fish. So I think when you think about high-throughput screening assays, if you understand the assay technology, the compound library, you might be able to work the same type of mathematics to give you a better chance of working on compounds that actually make biological sense. So here's how we did that with this program with the Charcot-Marie Tooth Association. So CMT1A is a peripheral neuropathy that's due to a gene duplication in the PMP22 gene. And this is what I'm working on. So for some reason, an extra copy of PMP22, it's a myelin structural protein, causes the myelin to degrade and changes nerve conduction. And I don't understand the physiology of this, but I was asked to work on it, and I have some good collaborators. And what we've decided to do is develop a reporter-based assay. And I won't go into the details, but we're basically building a construct around a neutronic element, which John Sfarron at the University of Wisconsin identified as a strong SOX10 EGR2 binding site within the gene. And we're using that to basically drive either a firefly luciferase or a beta-lactamase reporter. Now we've made two cell lines, one that contain each of these reporters. And the idea being that using the right type of cell line that overexpresses PMP22, here we're using a rat S16 cell, we have what I call orthogonal reporter pair. And these are cross-validating pairs. So we want to be able to see a downregulation in firefly luciferase activity, but also in beta-lactamase. And as a counter screen, we have cytotoxicity with a cell Kleider glove. Because since we're looking for decreases in a cell-based assay, that could be due to over-toxicity of the cell line. So this has got to be negative, and these two have to be positive. They both have to be positive. And so I'm not going to go on dwell on the nature of these reporters. You probably know about bioluminescent reporters or fluorescent-based reporters. You could read up on that if you're interested. This kind of assay, of course, can look for targets anywhere in the gene regulation mechanism. So we're coupling this. Go back. We're coupling this with our high-throughput screening we call quantitative high-throughput screening, where we look at a titration of the chemical library. So a five order of magnitude change in concentration across each sample. That gives us data that look like this. And we can do this for up to 400,000 compounds. We're combining the output of two different simultaneous reporter gene assays, and only when we see concordance between the two, for example, this middle pair, we would follow up the compound. These would be negatives, okay, because they have to report in both. And then we use a counter screen for firefly luciferase. So the data for this might just look like this. I'm going to run over, unfortunately. So the experiment goes 3,000 approved drugs go through both assays. We have criteria. Here are the actives. You can see there's hundreds, a few hundred, 120, 200, sorry, I can't even read my own things, about 129 actives, 144 or so actives across these two assays. However, the intersection is very small. It's only 34 compounds, shown here if you plot the IC50 of both assays. They fall on a nice 45-degree line, which is good. And then when we throw the counter screen in, we actually reduce this down to 9 compounds. Now, we want to have only a few compounds at the follow-up because we're using quantitative PCR to look at the message level of PMP22 in these cells as well as Westerns. And we can't really do that on hundreds of compounds. So here are some of the results. Again, in the end, what we end up finding are 3 interesting drugs that are able to reduce PMP22 levels. I would just point out bortezomib came out of this screen, and we've subsequently identified, a paper in the literature that kind of supports this. But the interesting thing is bortezomib, if those of you who know about it, it has a curious side effect. It has it's known as BIMP, bortezomib-induced bortezomib-induced neuropathy, peripheral neuropathy. And this, of course, is an anti-cancer agent. And it can create a neuropathy in those patients. So it's actually quite interesting that we found it as a molecule that reduces PMP22 expression. And so this might be an example of one of these Paracelsian type drugs where its off-target effect, its off-target toxicity in cancer might be an answer to dealing to treating CMT, maybe in a low-dose format. Now, so the last a case study I want to bring up, because Kurt will then build on this a little bit, is a biochemical assay here for protein-protein interactions. And this is, again, the tau fibrillation assay. And it's interesting, it's got two components. We have thioflavin T binding to polymerization of tau in the presence of heparin. So it's used to induce this polymerization or fibrillation. And then we have a substoichiometric amount of the labeled tau, which would then incorporate into these fibrils. And as the fibrils oligomerize or form these multimer complexes, we can use fluorescence polarization, which measures the rotation, the change in fluorescence polarization of a molecule as it slows down its rotation. So as the oligomers get bigger and larger, they'll be moving more slowly, and the polarization will change. So there's two outputs here. And just to throw the validation up here, the assay validation, and then the screening data, and I'm going to just focus you in on the some statistics. So for one of the thioflavin T binding component, we measure the Z factor of this assay. This is a statistical measure that allows us to look at the difference between the signal to background, between unpolymerized and polymerized material, and the standard deviation around the background and the signal. And through some statistics, one can get measures of the robustness of the assay. Anything above 0.5 is good. So we have a very nice, robust assay here. And just to show you some of the results from the screen. So the large screen we looked at the time, this was done a few years ago, 291,000 compounds. That was the size of the collection. That was equivalent to almost 2 million wells, because remember, these are titrations, and about 6 million outputs because of the nature of the reads we're making. And so here's just some of the data. If we look at the highest quality curve classifications, class 1, 2, and 3, I showed this earlier, we can parse out the molecules that are giving a pharmacological effect in both the THT and the MP assay by using a number of criteria that are listed on this slide. We were able to, of course, we can generate nascent SAR when we have this kind of information. We were able to identify 42 series that didn't have major liabilities, which are listed here, for example, promiscuous aggregation. And a number of the compounds were previously identified in other assays of this nature that were conducted. And in this particular example, we find an interesting new chemotype that had some a novel scaffold and promising drug-like features, and it showed good biological properties. For example, no significant effect on tau-mediated tubulin polymerization, which was something that the program did not want. So I think I'm going to end there. I'm just going to point out that high-throughput screening is really in the midst of translation. A lot of work needs to be done up front to develop the appropriate assay, choose the right libraries. I think we've heard about this. And then, of course, there's a lot of follow-up that occurs, so it's good to get the right compounds at the start. I want to just mention briefly that the NIH has a program that started about 8 years ago to do high-throughput screening. And so investigators would come to us with their assays, and we would do this, what's in blue. This was part of the Molecular Libraries program. We now have an assay development component. That's what I'm doing now. And then we have something known as TREND, Therapeutics for Rare and Neglected Diseases, which will actually progress compounds further to the IND. And these compounds can come from anywhere. They don't have to come from the NIH program. So this is an interesting, if you're interested, you might want to check out that website. And this is all now part of the new center at NIH. Some of you may have heard about it, the National Center for Advancing Translational Sciences. This was something Francis Collins wanted badly and was able to get Congress to agree to. So now I belong to this new center at NIH, and I think I'll leave it there. Thanks.
Thanks, Jim. I think we have time maybe for about
productivity. Right, so we work with a lot of different biology. And it's really the investigators that come to us, or the experts in biology. And the way the program is set up now, and maybe this wasn't clear, but we screened that large library of 400, it's now 400,000 compounds. All the data from that library, well, those screens go into something called PubChem, which is a repository that anybody in this room can access. And we happen to know that pharmaceutical companies use it on a regular basis. Why not? That's partly why it's there. And that kind of information can be derived from that database. So, with any specific biology, you can understand what the likelihood of finding some activity in our library is. I don't know for kinases, for example, what the percentage of activities are. I would imagine there's quite a few because there's targeted kinase libraries built into that collection. The idea, of course, for us is we would like to find, we take any type of assay, and actually we try to stay away from those targets that are commonly studied or that are studied to the best of our knowledge by pharma, because we want to look in areas that they're not looking to maybe help explore new biological, these undruggable targets, for example. Can we gain access there? But I don't know if I answered your question, but.
So there's a lot of discussion about mechanistic versus phenotypic screening,
Yeah.
and I'm curious because I've heard that whole organisms like C. elegans have been used for high throughput screening. So can you comment on the potential for scalability for those kinds of like C. elegans whole organism, or Drosophila, you had a picture of that in one of your slides, versus the cell-based models versus just more biochemical assays?
So this system that's set up currently at NIH is incompatible with C. elegans because the worm we just probably couldn't get enough of them into one 1536 well plate. However, I recognize that as a key organism for doing, and zebrafish as well, for doing screening. And we're working on basically backing off from the 1536 well paradigm and then using smaller targeted libraries to address those model organisms. That's something a lot of people are doing in academia, and we're going to explore that as well. But in this current incantation of the program, you really have to have something that's either a cellular-based assay, a cell monolayer we can grow, suspension, or a biochemical assay.
Any additional questions?
Hi. You addressed the question of titration
Yes.
of the compounds. What about the time factor? For how long do they need to be applied in order for you to see an effect?
Sure. Well, so kinetics are another tricky parameter to build into an assay. Some of them we can do. For example, an assay that's continuous in nature, for example, where there's a non-destructive change that we can measure over time, we can do that if we can look over the course of minutes or seconds, or hours if we can bring the plates back and forth. However, many of these assays are endpoint based, so to do a time course is very difficult. So that's one of the things we have to settle on as a fixed parameter. But we're doing a lot with respect to imaging, combining technology to use laser cytometry to do preliminary evaluation, and then look at microscopy to get more detailed analysis of what's going on with just those active wells. I think we can do similar things with kinetics. So the idea of the NIH is really to kind of innovate around the HTS paradigm. We all realize it's very imperfect, and we're trying to improve it. And the titration-based screening was one idea we had to do that.
Okay, if no other questions, thanks, Jim. Did you have extra slides at the end of your talk, Jim? Yeah, okay. I'll just work my way through them then. Oh, here's a C. elegans. Okay, so we're going to move on now to my presentation, and it follows on hopefully naturally from Jim's presentation, and essentially what is it that you do after HTS to further optimize compounds. And when I say further optimize compounds here, I mean beyond the kind of thing that most biologists think about, which is potency and perhaps even selectivity, but looking at some of these other important aspects of drug discovery that have been mentioned earlier today, such as ADME and toxicity. And so what I'd like to accomplish today essentially is to discuss what I think are certain systems and assays that a reasonably sized academic group, for example, could put into place to aid in this drug discovery process, to aid in the optimization of these other dimensions of drug discovery besides potency and besides selectivity. And so because each assay has obviously a different target by which you have to measure the potency and then related targets for which you want to measure selectivity, I don't have time to go through all those permutations today. Obviously, this is going to be target specific, and each group has to build these assays. Jim touched on the assay development aspects that would be required for HTS. Of course, you also need develop the assays for secondary analysis of selectivity. But there are certain aspects of the process, I think, that are kind of universal that, again, many academic labs should be thinking about and should put into place. And so I'll focus on the systems that can be put into place to look at ADME. And for those, again, who have forgotten the acronym, that's absorption, distribution, metabolism, and excretion. In other words, how the drug is handled by the animal. And related to these things are compound solubility, the pharmacokinetics, the behavior of the drug in the animal, how rapidly it's cleared, and drug metabolism, what enzymatic systems are involved in this clearance. And then I'll touch briefly on what I'll call the preclinical toxicology, the non-GLP type, that again